Since all living organisms pass along and inherit traits in basically the same way, the work of Mendel and other geneticists that began with the study of experimental subjects such as pea plants and fruit flies yielded powerful insights about the inheritance of traits that could be applied to humans as well as other living organisms. Through the work of geneticists, we now understand that hundreds of human diseases don't result from a cause in the external environment, but are rather the result of genes inherited from one's parents. However, genetic research has also made us aware that our predisposition to hundreds of other diseases, such as heart disease and cancer, is the result of a complex interplay between the genes we inherit and other external factors, such as our personal habits and the environment that surrounds us. Let's now take a look at some major recessive, dominant, and sex-linked single-gene disorders, disorders caused by inheriting too much or too little genetic information, and some human traits that are controlled by multiple genes, before looking at some of the techniques used by genetic counselors and others to provide couples with information about the likelihood of passing various genetic disorders to their children. Cystic fibrosis results from the failure of proteins in cell membranes, which allow chloride ions to pass out of the cell. The inability of the chloride ions to cross the cell membrane properly also affects the transport of sodium ions and of water in and out of the cell. The result is that cystic fibrosis victims, rather than having the thin, free-flowing mucus of most individuals, have mucus that becomes thick and sticky as a result of the lack of water in the mucus. About 1 in 20 Caucasians carries one of the recessive alleles for cystic fibrosis, and this results in 1 in 2,000 Caucasian babies being born with cystic fibrosis, which translates into 4 or 5 babies born with the fatal disorder each day in the United States. In recent years, the knowledge of cystic fibrosis and advances in medicine have improved the quality of life for people with the disorder and substantially lengthened their life expectancy. This is the result of physical therapy, breathing exercises, modern antibiotics, and special diets. But eventually, the disease does prove to be fatal. Sickle cell anemia results from just a single amino acid being different in the polypeptide chain that forms hemoglobin molecules. This slight change causes the abnormal hemoglobin molecules to be less soluble and form crystals within red blood cells. The crystals distort the red blood cells into a sickle shape that gives the disease its name. In addition, the formation of crystals prevents the hemoglobin from carrying oxygen effectively and makes sickle cells rigid so that they often block capillaries. Blocked capillaries can lead to pain, fever, and weakness in affected areas. Clots formed by sickle cells can break off and cause stroke in even very young children. Like cystic fibrosis, the life expectancy of sickle cell patients is being substantially lengthened as a result of a variety of therapies, including transfusions, antibiotics to treat infections, and drugs that increase the oxygen-carrying capacity of the red blood cells. Sickle cell anemia currently affects about 1 out of 500 Americans of African descent, while about 8% are heterozygous carriers of the disease. In parts of equatorial Africa, 1 out of 25 individuals have the disease, while about 30% are carriers. Given the severity of sickle cell anemia, one asks why natural selection hasn't eliminated the sickle cell alleles. The answer may lie in the fact that being heterozygous for the sickle cell trait affords protection against an often fatal form of malaria. This form of malaria, like all forms of the disease, is caused by protozoans carried by mosquitoes that invade an infected individual's blood cells. Abnormal hemoglobin, which accounts for about half of the hemoglobin in heterozygous individuals, appears to hinder the development of the disease-causing protozoans in an individual's blood cells, and thus often turns out to be the difference between life and death in heterozygous individuals infected with this form of malaria. In most other regards, such as health and physical prowess, the 50-50 combination of normal and abnormal hemoglobin appears to leave heterozygous individuals largely unaffected. In fact, many world-class athletes are heterozygous for the alleles that cause sickle cell anemia. Being heterozygous for the gene responsible for Tay-Sachs, a fatal disease affecting the nerve cells of infants and young children, has been hypothesized by some researchers to provide protection against tuberculosis, in much the same way that being heterozygous for sickle cell anemia protects against a form of malaria. Until recently, tuberculosis was a very substantial threat to people living in closed urban environments in Eastern European cities, where Jews made up a significant percentage of the population. The death rate from tuberculosis was as high as 20%. However, when genetic researchers looked back at the death records for the grandparents, of whom at least 50% had to be heterozygous for Tay-Sachs, only 1 out of the 360 studied had died of tuberculosis. This led to the hypothesis that perhaps the same qualities that led to Tay-Sachs disease in a homozygous individual lead to protection against tuberculosis in a heterozygous individual. Infants homozygous for Tay-Sachs are unable to make an enzyme critical to breaking down a lipid produced by nerve cells. With no enzyme to break down these lipids, they eventually build to toxic levels. As a result, Tay-Sachs infants who appear normal at birth over time begin to suffer convulsions, loss of motor control, blindness, and loss of contact with their parents and the world. Children born with Tay-Sachs rarely survive beyond their 4th or 5th birthday. Among the population in general, 1 in every 400,000 children are born with Tay-Sachs. But among Eastern European Jews and their descendants, the disorder historically affected about 1 out of every 3,600 births, or about 100 times more than the non-Eastern European Jewish population of the world. Fortunately, screening procedures that measure the level of the critical enzyme in likely carriers now provides most of those at risk of having a child with Tay-Sachs with relevant information regarding the decision to have children. All of the genetic disorders we've looked at so far are recessive. That is, they remain hidden if an individual also inherits a properly functioning dominant or codominant form of the particular gene. Thus, heterozygous parents carrying these illnesses have no symptoms. But what if a genetic illness is inherited via a dominant allele? One of the parents must have the disease. Genetic illnesses carried via dominant alleles that are fatal at an early age or so severely disabling that they make the individual undesirable for mating die out immediately in a population. The one major genetic disorder carried via a dominant allele found in the human population today is Huntington's disease, which has continued in the human population in spite of the fact that it is both disabling and ultimately fatal to those who inherit it. The reason Huntington's disease has persisted in the human population is that individuals afflicted with it appear totally normal until their 30s, 40s, or even 50s, and thus have often had children long before having any symptoms of the illness. Unlike the recessive disorders we've looked at, scientists are still unable to explain the protein, enzyme, or other molecular mechanism through which Huntington's disease occurs. But they have identified the physical change in the brain, a tremendous enlargement of structures called ventricles that cause the symptoms of Huntington's disease: loss of coordination, flailing movements, personality disturbances, and eventual death. The American public first became aware of Huntington's disease in the 1950s when the folk singer Woody Guthrie, who wrote "This Land Is Your Land" and many other classics, started developing symptoms of the disease. In 1993, Dr. Nancy Wexler of the Heredity Disease Foundation initiated field studies delving into the causes of Huntington's disease in Venezuelan villages near Lake Maracaibo, where as many as 1 of 5 individuals are afflicted with Huntington's disease. Wexler sent blood samples back from the field to Dr. James Guisella and his colleagues at Massachusetts General Hospital in Boston for molecular study. This coordinated effort by Wexler and Guisella resulted in the discovery of the exact location of the gene responsible for Huntington's disease, on the tip of chromosome 4. Further research determined the exact DNA sequence for various alleles of the gene. One of the immediate results of their finding was the development of a more accurate test for detecting individuals who will become afflicted with Huntington's disease. The alleles of the gene that cause Huntington's disease are substantially different from normal alleles of the gene. In an expandable region of the gene that is usually no more than 60 bases long, the Huntington alleles are greatly expanded to as many as 150 bases in length. Determining the exact sequence of bases in the defective alleles has allowed researchers to compare the protein produced by individuals afflicted with Huntington's disease with the proteins of non-afflicted individuals. There is hope that understanding the different forms of the protein produced by the gene at the tip of chromosome 4 can lead to the development of an effective therapy for Huntington's disease. Meanwhile, the development of an accurate test for Huntington's disease has created a tremendous dilemma for individuals who are at risk for inheriting the fatal gene: whether or not to take the test. Having a parent afflicted with Huntington's disease means one has a 50-50 chance of inheriting the disease themselves. A 50-50 chance of hearing that they have inherited a terrible and at this point always fatal disease. A 50-50 chance of hearing they've escaped a genetic curse. Only those faced with such a situation can understand the depth of the dilemma. Now we will look at 3 other recessive single-gene traits: color blindness, hemophilia, and muscular dystrophy. These are like the other traits, except the genes for these traits are sex-linked because they are found on the X chromosome rather than the normal autosome like the other traits. This affects significantly how they are inherited. All sex-linked genetic disorders tied to the X chromosome occur at a substantially higher rate in males than in females. Remember that the Y chromosome is much shorter than the X chromosome, and thus lacks many of the genes found on the X chromosome. Thus, all sex-linked alleles are expressed in males even when they are recessive, as there is no second complementary gene which can have a dominant allele that will cover up the recessive gene. Females have 2 X chromosomes, and thus have to have 2 recessive alleles of a gene to inherit a recessive sex-linked genetic disorder, which is much less likely than inheriting a single recessive gene. A classic example of this is red-green color blindness, in which affected individuals have difficulty distinguishing the lighter shades of red and green because the cones of their eyes can't synthesize the pigments required to distinguish these colors. In Caucasian populations, about 8% of males have the disorder as compared to about 1% of females. Hemophilia is another recessive, sex-linked genetic disorder which often was associated with royal families of Europe, particularly descendants of Queen Victoria of England. Hemophilia is the result of a deficiency in one of the blood-clotting proteins. The most common form of hemophilia, hemophilia A, results from a deficiency of clotting factor 8. Individuals with hemophilia can bleed to death from minor cuts and may suffer internal bleeding from bumps or bruises. Fortunately today, hemophilia can be treated by injecting a clotting factor isolated from donors' blood. Unfortunately, there was a period of time not too long ago when many hemophiliacs contracted HIV as the result of injecting clotting factors isolated from HIV-positive blood. Fortunately, today, there are better blood screening devices, and recombinant DNA technology promises the ability to produce clotting factors made from non-human sources and produce it at much lower prices. Hemophilia is a relatively rare disorder, affecting only one male in 10,000 and one female in 100 million. In other words, there are 25 to 30,000 male hemophiliacs in the United States and only 2 or 3 female hemophiliacs. A third sex-linked genetic disorder is muscular dystrophy that results in the progressive wasting away of skeletal muscles. The most common form of muscular dystrophy, or MD, is caused by an allele that causes a defective form of the muscle protein dystrophin to be formed. Researchers are currently looking at ways to insert the proper alleles of the dystrophin gene into muscle cells via a technique called gene therapy. One interesting phenomenon should be mentioned about all these sex-linked traits. While males predominantly inherit the trait, males can only pass it on to their daughters. That is because these traits are located on the X chromosome, which is the chromosome which in a sperm caused the formation of a female embryo. Thus, sex-linked traits tend to skip a generation, as fathers pass it on to a daughter, and that daughter, in turn, passes it on to a son. Not all of our traits are controlled by the inheritance of a single gene. Our height, weight, and the color of our eyes and skin are all controlled by more than one gene, and are thus referred to as polygenic traits. That eye and skin color are controlled by more than one gene, respectively, is reflected in the fact that there is such a variety of gradations of eye and skin color. People don't just have blue eyes and brown eyes, but rather there are blue eyes, green eyes, hazel eyes, light brown eyes, black eyes, etc. The same is true for skin color, and perhaps has even more variations. Both eye color and skin color are the result of the presence of a pigment called melanin. If melanin production were controlled by one gene, and an individual had 2 recessive alleles and no melanin was produced, they would have light skin. If they had one dominant gene and one recessive, their skin color would be of medium tone. And if they had 2 dominant alleles, they would be extremely dark. However, we see a much greater variation in skin color than this among individuals. This is the result, biologists believe, of at least 3 incompletely dominant genes controlling skin color. These combinations among 3 different genes allow for 7 different shades of skin among individuals. Likewise, biologists believe 2 or 3 genes control eye color, explaining all the various shades of eye color, as opposed to just one gene controlling eye color, which would result in only 3 different eye colors, say blue, light brown, and brown. But as we know, there is a much wider variety of eye color than this. There are also a large percentage of infants who have blue eyes that later become a darker shade, such as hazel or brown. And this is probably due to the fact the infants have not started producing melanin in their irises yet. There are a number of disorders that result not from inheriting defective alleles for a gene, but rather as a result of inheriting too many or too few chromosomes. As you will recall, during meiosis, the different homologous pairs of chromosomes separate to form either sperm or egg. Occasionally, a chromosome doesn't divide properly, and both members of the pair go to one of the gametes, while the other gamete is left without a chromosome at all. Such errors in meiosis are called non-disjunction and can arise in the distribution of both sex chromosomes and autosomes. Most embryos that arise from the fusion of gametes with abnormal chromosome complements spontaneously abort, accounting for between 20 and 50% of all miscarriages. But some survive. Non-disjunction can also lead to abnormal numbers of autosomes. Fusion of a normal gamete with an abnormal gamete can lead to either 1 or 3 copies of an affected autosome. If an embryo receives only 1 copy of any autosome, the embryo aborts so early in development that the woman never knows she was pregnant at all. When there are 3 copies of a given autosome, that usually also causes spontaneous abortion, but often later in pregnancy. However, some trisodometic babies are born, especially those with 3 copies of either chromosomes 13, 18, or 21. Of these, trisotomy of chromosome 21 is the most common and produces what is known as Down syndrome. About 1 in every 800 children born in the United States has Down syndrome. Down syndrome results from receiving 3 complete chromosome 21s, or rarely from having 2 chromosome 21s and a small part of another chromosome 21 attached to another chromosome, often chromosome 14. Down syndrome can result in mental retardation that ranges from mild to severe. Typically, only about 1 in 25 children with Down syndrome ever learn to read, and only about 1 in 50 learn to write. Down syndrome is recognizable by a number of distinguishing physical traits that include short stature, a lack of muscle tone, a mouth held partially open because it cannot accommodate the tongue, and distinctively shaped eyelids. Many individuals with Down syndrome also exhibit low resistance to infection, disease, and are born with heart malfunctions, vision problems, and intestinal problems. Individuals with Down syndrome also have an increased risk of developing a cancer of the blood cells known as leukemia. 60 years ago, most people with Down syndrome died before the age of 10. Now, with better care, the average lifespan is increased to 30 years. About 25% of individuals with Down syndrome now live to be 50. The frequency of all types of non-disjunction increase with the age of the parents, especially the mother. After the age of 37, the rate at which non-disjunction and Down syndrome occur increases tremendously. As a woman approaches the age of 48, 3 out of every 100 births result in an infant with Down syndrome. Biologists hypothesize that this occurs because, as you will remember, meiosis and the production of eggs begins in a woman while she is still a fetus in her own mother's womb. Thus, the eggs of a 40-year-old woman are over 40 years old. The process of aging, exposure to radiation, toxic chemicals, and viruses all increase the likelihood of non-disjunction occurring as meiosis is completed in a 40-year-old egg during the monthly menstrual cycle. Men, on the other hand, produce sperm cells continually. But nevertheless, 25% of cases of Down syndrome can be linked to non-disjunction in the sperm. Just as couples can be given a probability about the occurrence of Down syndrome, they can also be given a probability that one of their children will inherit a given single gene disease, such as cystic fibrosis or Tay-Sachs, by having a genetic counselor take a family history and then use the information to predict the probable appearance of a disease in their children in much the same manner Mendel would have predicted the probable distribution of traits in a given generation of pea plants. In the case of certain genetic diseases, diagnostic tests can be carried out to determine if one or both parents is heterozygous for a trait and therefore a carrier. For example, black couples that are suspected of being carriers for the trait of sickle cell anemia can have their DNA tested. Likewise, fetuses and infants can have their DNA tested to see whether they are heterozygous for the sickle cell trait or homozygous, which would produce the severe symptoms associated with sickle cell anemia. Likewise, Jewish couples that might appear to be at risk for being carriers of Tay-Sachs disease can also be tested to see if they are carriers. However, in this case, it is not the DNA that is tested, but rather the level of the enzyme that is to be produced by the gene that is tested. Carriers for Tay-Sachs disease typically have 1/2 the level of the enzyme produced by this gene as a typical individual would have. If couples were found to be carriers but decided to go ahead and conceive anyhow, the fetus could be tested for one of these genetic conditions via two methods: amniocentesis or chorionic villus sampling. Amniocentesis involves inserting a long, thin needle through the waterproof membrane called the amnion, which surrounds the fetus and holds the amniotic fluid. The amniotic fluid contains fatty acids, steroids, free amino acids, enzymes, and other proteins, as well as cells. The chemicals come from both the mother and the fetus, but the cells all come from the fetus as a result of being shed by the skin and digestive tract or respiratory tract. After a fetus becomes 16 weeks or older, enough amniotic fluid has accumulated to provide the room necessary for the long, thin needle to be inserted and take a sample of the amniotic fluid. Before inserting the needle, a physician determines the position of the fetus by ultrasound and then inserts the sterilized needle through the abdominal wall of the pregnant woman and into the amniotic fluid. Usually, a small amount of fluid, approximately 10 to 20 milliliters, is withdrawn. Biochemical analysis for enzymes and other proteins can be performed on the fluid immediately. However, there are typically very few cells in the fluid sample, and to perform various procedures that involve looking at the genes of the cells must first be grown in culture. It usually takes a week or two to acquire enough cells for a process called carrier typing, or the type of analysis of DNA used to determine whether an individual has sickle cell anemia. Chorionic villus sampling is a newer procedure that involves inserting a small tube into the uterus and suctioning off a few villi from the chorion, which is the membrane that eventually becomes part of the placenta. The cells that make up the chorionic villus are entirely fetal cells. Chorionic villus sampling has two major advantages over amniocentesis. First, it can be done much earlier in the pregnancy. The second advantage is that the sample contains a much higher concentration of fetal cells than in amniocentesis, so analysis can be performed immediately rather than waiting a week or two. However, chorionic villus sampling appears to have a somewhat higher risk of inducing miscarriages than does amniocentesis, although the risk is small in either case. A second disadvantage of chorionic villus sampling is that the chorionic cells seem to be more likely to have abnormal numbers of chromosomes, even in a normal fetus, than do the cell samples by amniocentesis. This complicates the diagnosis of abnormalities due to abnormal numbers of chromosomes, like Down syndrome. Which procedure is chosen undoubtedly depends on the objectives of the procedure and what the doctor and patient feel most comfortable with. While amniocentesis and chorionic villus sampling certainly provide couples with information, they can't answer the tough ethical, moral, and emotional issues they often leave a couple facing. While we looked at a number of striking genetic illnesses that are caused by inheriting one or two abnormal alleles of a single gene, or an incorrect number of chromosomes, we are all more or less susceptible to conditions such as heart disease, diabetes, cancer, schizophrenia, or manic depression based on the genes we inherited from our parents. But for most of these conditions, the advice of a genetic counselor would currently be of little use, as these disorders are caused by a complex interplay between numerous genes and external factors, such as diet, exercise, smoking, drinking, drug use, and social and family dynamics. Nevertheless, there is every reason to believe that in the future, researchers will slowly unravel the mysteries of the human genome, and with the information they obtain, will become increasingly able to predict and treat a wide variety of genetic illnesses.